Yesterday, we talked about consanguineous mating, not consanguineous mating. Sorry, guys, I've been saying that wrong since I learned it. But we were talking about consanguineous mating and how it makes specifically female research mice extremely unreliable in preclinical trials. And in that video, I got this comment that I absolutely love. This is the entire comment, but it basically says, this isn't my field of expertise. I'm a biologist, but not a geneticist. However, mammals undergo something called X chromosome inactivation, which is a process of silencing half of the genes randomly so that there are only one functional copy of each gene in the somatic cells. Essentially, all females only have one active set of X chromosomes. While I'm sure the lack of genetic diversity could have some impact, it is not different than the impact that is seen in every other trait study for this reason. There needs to be much more research on how medications impact females, absolutely no doubt. But this isn't a main root of that issue. The fact that mice are inbred allow us to know exactly what genes we are working with, which is incredibly valuable information. Now, when it comes to this comment specifically, I do agree. For one, we definitely need to do much more research on medications regarding female humans. And two, I really appreciate the mention of X chromosome inactivation. However, it is important to note that there is an aspect of X chromosome inactivation that does make it matter as to whether two X chromosomes are genetically diverse or not. Let's start with what X chromosome inactivation is. You see, whenever you have two or more X chromosomes, one of them has to be inactivated, like the comment said. So it undergoes this thing called X activation, where RNA, called exist RNA, basically wraps around one of the X chromosomes. Now, how it inactivates it is the exist RNA basically wraps around the chromosome so tightly that nothing can really get to the genes on that chromosome. And this tight packing of this chromosome thus inactivates the X chromosome and turns it into what we call a bar body. Now, you probably learned that in basic biology, the basics of X chromosome inactivation. However, what a lot of basic biology courses don't cover is that whenever an X chromosome is inactivated by the exist RNA, causing it to turn into a bar body, not 100% of the X chromosome is actually inactivated. In fact, whenever an X chromosome is inactivated, around 15 to 20% of the genes on that X chromosome are still able to be coded, thus causing genetic diversity between two X chromosomes in an individual to still matter whenever it comes to research results, as that 15 to 20% of the genes that are still able to be coded and active do make a difference in research.